All right, I see people are starting to join. I think we'll just give it a minute or so and see if any more join us, and then we will get started. Maybe give it 1 or 2 minutes. All right, I think I'll get started with some of the housekeeping items that'll get us going. And firstly, I'd like to introduce myself. I'm Bill Farrell. I'm at Pfizer in La Jolla, California. I'm an associate research fellow, and I'm one of the team that developed the topic that we're talking about today, the EPSA assay. And we had worked on this assay as originally for small molecule, and it seems to have really found a niche for large molecules, and peptides. So I think we're going to have a very exciting event today. And I want to welcome everyone, either good morning, good afternoon, or good evening, wherever you are in the world. I know this is unprecedented times to be using these forums so much these days. Hopefully this will be a great event, and it's great that we can reach so many people this way. I also want to thank Averica for bringing together this team, this panel today, to go over this assay and to open it up for a question and answer session at the end. The first bit of housekeeping is that the event is being recorded. So this will be found on the Averica website after the presentation, but be advised that we are recording this. We will have an open panel discussion and followed by a Q&A. And please use the Q&A function at the bottom or on the side of the screen in the Zoom controls, not the chat function. We'll be monitoring the Q&A. And I think with that, we will get started. Today we have our featured speaker, my colleague from Groton and my collaborator in all of this, the years really of working on this assay, is Gilles Goetz, principal scientist at Discovery Sciences in our Groton, Connecticut facility. Gilles has been an outstanding collaborator and really drove the assay for peptides, and working with another member of our team, put out the publication I assume that most of you have seen, otherwise you wouldn't be here. It's been really a pleasure working with him. And so I guess I'll turn it over to Gilles to get us started on for some presentations today, and then we will hear from several panelists. I just want to briefly go through who they will be. We have Romulo Romero. He's an analytical chemist, over 20 years of pharma discovery experience. He's currently focused on SFC, and he has interests in automation, high throughput processes, as well as he's, as you can see from his background, he's eagerly awaiting the landing of Mars Perseverance today. So he's like chomping at the bit to get off and go watch that. We have Huy Nguyen, scientist from Analytical Research Group in Genentech. His group focuses on running phys-chem assays like EPSA, log-D, solubility. And in addition, Huy's group utilizes microfluidics to develop novel phys-chem assays. We also have Erin Jordan, who received her bachelor's degree from University of Minnesota and her master's in chemistry from Northwestern University. And she, while performing synthetic organic chemistry under the advisory of Carl Scheidt, Erin began her career at Abbott Labs in 2009, providing cryopurification support to process chemistry. In 2011, she transitioned to Abbott AbbVie Drug Discovery, where she currently co-leads the chiral SFC platform supporting MedChem therapeutic teams. Erin has also been running the EPSA within her groups for the last couple of years. Joining us is Johan Nicolai, a senior scientist in the DMPK department at UCB Biosciences. After obtaining his PhD, he joined UCB in 2016 and focused on the prediction of brain disposition of small molecules before moving to the area of intestinal drug absorption of new chemical entities. More recently, he's been involved in the evaluation of permeability of beyond rule of 5 compounds and is looking for ways to relate beyond rule of 5 compounds passive permeability to phys-chem parameters such as EPSA. And lastly, we were going to be joined by Paul Lefebvre from Verica to give a brief overview of the discovery services there. However, we're glad to say he's expecting his first grandchild today, so he's not going to join us, and I will fill in from there. I think with all that out of the way, we will turn it over to Gilles. You want to kick us off?
Well, thank you so much, Bill, for the great introduction. Let me see if I can still manage that sharing. Okay, so is it there? Can everybody hear me and see my slide? I assume yes.
Yes, I can hear you and see you.
All right, so I'm here to basically give you a short overview of what EPSA was, what EPSA is, and what EPSA has become in terms of what it came out of, which was SFC, and how it managed to become a polarity readout. Furthermore, enabling design, but because it was also a permeability surrogate. That's one of the key contribution of the EPSA. And just to be clear, it is EPSA. There's 4 letters. It is not EPSA. And Bill and I have been grinding our teeth each time we heard EPSA. So in your questions, we will read EPSA because you're going to type it. So what's the actual problem here? Well, the fact of the matter is that in the pharmaceutical industry, we want to make and produce and generate oral available drugs for all the benefit of getting an injection for a vaccine, for example. People are just more compliant and like just to take a pill in the morning and be done with it. And over here and in every lab that I know of in the industry, we are very good at making potent compounds. The fact of the matter is that a potent compound is not quite sufficient to make an oral drug. You need to have something that is permeable and that is water soluble as well. And that's where the EPSA found its niche. So an oral drug actually needs to cross the blood-brain barrier. Not the blood-brain barrier, but the sorry about that. That's a misnomer. It needs to cross the intestinal wall to enter the bloodstream before reaching its target. And those compounds need to be cell permeable, obviously. And some will be pumped in to active transport, but most will get in with passive permeability. And there's actually 3 key properties that will actually contribute to the passive permeability. Lipophilicity is one of the first one, and it's typically described using log D, log P, or the calculated version, C log P. Then you have the size of the compound, of the molecules that affects the diffusion of the membrane. Typically, you describe the size using molecular weight or hydrodynamic radius. And then you have the one that we are actually interested in, polarity. And the polarity is an estimate of the free energy barrier associated with the dissolution of hydrogen bonds upon insertion of the membrane. Typical descriptors of polarity are TPSA, hydrogen bond donor count, hydrogen bond acceptor count, and the one that we are really interested in here today is EPSA. The hypothesis we work under is that in order to be permeable, a compound needs to hide its polarity. Why? Because of that dissolution penalty. And you can see it in the bottom. Inside of a membrane, intramolecular hydrogen bonds are favored, and a compound like the one described or shown here is actually going to be able to shed the water molecule that were all around him when it was in the outside in solution, and be able to favorably have that intramolecular hydrogen bond that will allow him to come out. And those compounds can or have been called chameleonic compounds recently. And again, the key here is that intramolecular hydrogen bonds are favored inside of a membrane. So how can we take advantage of that? So the EPSA here, which has been developed using our expertise in SFC, is based on a Carex 3014 column. And we picked that column because we tried a whole lot of them. And then we realized that there was a lot of similarities between Carex structures and what you see in a bilayer. You're not going in this direction, you're going in the other direction. But in essence, what you use is the availability of that SFC technique that is going to generate a polarity readout, because there is polar-polar interactions inside of that membrane into those conditions. And we obviously generate a retention time. And the key here is that the supercritical conditions of CO2 and methanol that is used in our conditions generate a dielectric constant that is spot on comparable to what you have inside of a membrane. In essence, you are in the same typical conditions that are favoring intramolecular hydrogen bonds, as shown here. So obviously, a polar compound in those conditions is going to retain longer in SFC. And if you have an intramolecular hydrogen bond that can be made, that can be formed, it is going to be formed. It's going to generate a conformational change. It's going to reduce the polarity of the compound. And by extension, it's going to diminish its retention on the column. So we're working with medicinal chemists. And those medicinal chemists, when they think polarity, they think TPSA, which is an acronym for topological polar surface area. That polar surface area, in essence, is just an addition of contributors for every single compound or every single polar bits of the compound. It's just a list, you add it, and the flat structure can give you a TPSA. What we did here is that we generated SFC retention times for a set of 120 or so vanilla compound. We call them vanilla compound because they will not have any intramolecular hydrogen bond. They're just well-behaved compounds. And what we were able to do is build a descriptor operating in the same scale as the TPSA. So we basically took TPSA values of those 120 compounds and generated the retention time in our assay, and then came up with a correlation, right? Where you have R square of 0.94 here. And after that, in order to make that compound, that assay portable, we calibrated it using a list of compounds shown here, which were spanning the retention time. So between 2 and roughly 14 minutes, extending further out than the one over here. And we can generate then and assign an EPSA value for each and every single one of them. And then what we do is we keep those EPSA values because those are the standards. And then every time you make another analysis, you may have a different retention time. But that retention time is no longer relevant because we have those EPSA values. And therefore, you back calculate a new correlation where you have A times retention time plus B equals EPSA. And that's how you can actually generate your values every single time. So that was a long technical explanation, but what actually comes out of it is what we can see here. You have here two compounds that have the same structure in essence. They are just regioisomers of each other. The piece here is then just off by 1 carbon. It has the same individual bits. It has the same total topological polar surface area, which is calculated for those two regioisomers. But what happens is that you can see that the one in blue has a longer retention time than the one in red. What happened is that the one in red actually has an EPSA value of 106 compared to the one in blue, which has an EPSA of 119. And the fact of the matter is that the difference here can highlight the fact that indeed you can form an intramolecular hydrogen bond over here. And typically what we found is that if you have a difference in polar pairs, like you can see here, molecular match pairs, you can suggest having an intramolecular hydrogen bond with an EPSA over 3. That's for small molecule. And if you now look at larger molecule, you don't necessarily need those molecular pairs. So those two compounds here, they're not clearly matched pairs, but they are pairs no matter what. By all typical medicinal chemistry rules or medicinal chemistry knowledge, the one on the top here has less hydrogen bond acceptor and donor and less rotatable bonds. It's calculated TPSA is 105. If you compare it to the one at the bottom, has more hydrogen bond donors, more rotatable bonds, its PSA is 175. So you would expect that the one on the bottom is more permeable than the other, just because it is calculated to be more polar. Turns out that the permeability of the top one is lousy, and the permeability of the bottom one is really good. 19 times 6 centimeter per second is outstanding in terms of permeability. Well, it turns out that if you now measure the EPSA after the fact, you can actually see that the high EPSA actually tracks with the poor permeability and the lower EPSA tracks with the good one. What explains that? Well, I mean, I kind of gave it away earlier. It's all about intramolecular hydrogen bonding. And that compound 13 here can actually not even form one, but actually form 2 intramolecular hydrogen bonds and completely hide its polarity, making it a shoo-in to go through that membrane. So what happened is that after finding that out, all the following designs incorporated compound portions and bits that could actually join each other and form intramolecular hydrogen bond in the correct conditions. Now, let's move on into cyclic peptides. We looked at a whole lot of known peptides and found out that there is a lot of similarity here. A high EPSA seems to track systematically with poor permeability. So we had that starting point, and then we figured, let's see if the ones that have a low EPSA actually start being more permeable, and it is the case. Examples here of cyclosporine and one of the NME3 low-key compounds have quite reasonable or quite low EPSA and actually have very good permeability. We expanded that using a collaboration we made with the low-key group in California and found that plot here, permeability and EPSA. Looks like there is a magic number here. Looks like there is a key 80 value of EPSA that seems to be the one that you want to have. You need a low EPSA. I'm not saying that EPSA is going to be the necessary, the indispensable parameter here, but if you don't have low EPSA, you will not have anything permeable. See, that's what you can see on the right-hand side here. So we extended that study to a whole lot of different cyclic peptides and came up with the slide here now. You can see that if a compound has EPSA below 80, it seems to be quite reasonably permeable. If it is above 100, I mean, everything is red. There is no permeability. Below 1 is lousy. And above 5, in terms of permeability, seems to be quite reasonable. So what we recommend now is to measure EPSA, and if the EPSA is below 80, you can go ahead and measure permeability. You have a good chance of having something that you can read. And on the other hand, if the EPSA value is above 100, don't waste your resources, don't waste your precious cells and your time to measure permeability. Chances are you're going to be in the grass, not even able to measure it. And we can see that in the quite busy slide here. There's three projects that went with us into that learning curve in terms of EPSA and permeability. The first one, project in blue, started in the 116-60, and basically what they saw is that they could not get any permeability value, and so they could not basically see in what direction they were going. Is adding that methyl something important or not? Is it pulling me in the right direction? So we used EPSA to show them that indeed that methyl addition is actually getting you into the right direction. And it went to the right direction, but not sufficiently enough. So what they did is that they actually designed two specific compounds. Forget about potency for those. They just wanted to see if they can make it to the permeability barrier. And they pushed the lipophilicity and the polarity down in order to get it into below 80. And sure enough, they were picking up something that is measurable. The other two projects were able to piggyback on that learning and started faster and were able to bring EPSA down to that below 80. And sure enough, as soon as they were in below 80, they were actually in an area where they could measure permeability, and they could be in a permeability area where it is actually very favorable. So once you have a whole lot of measured values, you can actually leverage that information and generate a computational model. And that's exactly what we did. We are able to generate an in-house EPSA model that was used for design cycles. So basically what the designer can do now is design a virtual library, feed it into the EPSA model. If the EPSA model comes back with calculated EPSA or predicted EPSA below 100, they would go ahead and use the synthetic efforts to make that compound. Once it's made, you test its potency and its EPSA. And if it's potent and the EPSA is below 100, you can actually go ahead and verify that it's actually permeable or not. If it's permeable, you use those learning into the next design cycle, and that's how the project 3 was able to get into such a great space. This is just a review of the EPSA values and the number of compounds that had reasonable ROCK here, and it's exactly how you expect it. Now, just real quick, the project here, CXCR7, was able to come from the starting compound here, starting peptide that had a also potent, but their P up, which means their permeability was dreadful, with an EPSA of 149. After a series of design cycles, they were able to get something that was exquisitely potent with a very low EPSA that correlated with a very reasonable cell permeability that actually converted into an actual reasonable 18% bioavailability. So that's essentially how we were able to use EPSA to drive design. And now that I'm already done, I just want to give kudos to all the folks that contributed to the success of EPSA. And I'm gonna turn it over back to Bill and to our panelists. Thank you again.
All right, so I do have there were a couple questions that came in, Gilles, that I think are probably a little relevant, to kick off right now, and then we'll move on to the next panel speaker. So one was, did you ever try a phosphatidylcholine IAM column from Regis for intermolecular hydrogen bonding, as that's like a real lipid membrane?
We tried it, but it's not the greatest stability in SFC. I think what I remember is that it was not working the way we expected it to work. Probably because the silica was not designed for an SFC, and in order to have it done by HPLC, you're basically no longer favoring those intermolecular hydrogen bonds because you are in very aqueous media.
So I also remember, in answer to this question, years ago, we had actually had a custom column made, which was phosphatidylcholine as well as cholesterol. And we had that custom packed by our friends at Princeton Chromatography. And it looked about the same as the Kyrex column. However, it was expensive to make, custom, which means it was going to be hard to control. And so we just sort of walked away from it and went for something commercial. That's a great question. And the other one is, can you describe a bit more on how you select the standards, which includes a number of acids? And that was the question.
Well, those standards were picked essentially because they were commercially available and because they were spreading the breadth or the distance between a very short eluting compound and a very long eluting compound. We didn't we picked them because they were available, because you can buy them off the shelf, and because you have a one that starts very low and again all the way across. And if it's an acid, it just happens to be one of the reasons why it elutes on the longer time side of the elution profile.
Great. All right. Well, I'll put the rest off for right now. And
I actually have a question.
Go ahead.
I was wondering, so you showed a drop in retention time for when you see intermolecular hydrogen bonding, but one thing I've observed is it's a chiral column. So what if you have a racemate that's submitted and you'll have one enantiomer versus the other? Is it a similar kind of relationship, like intermolecular hydrogen bonding, where it's actually structurally dependent on where those polar groups are facing? Or how do you interpret those type of interactions?
Well, that could be one of the tricky pieces, and especially if you have one. I mean, I wouldn't think that the two stereoisomer would generate yes or no intramolecular hydrogen bonding. I would think that the positional location of those polar or donating or accepting groups would actually work in concert. So you might get a pair on before and a pair that later. I don't know if I'm clear here.
Aaron, if I may chime in, a couple of times I've run some racemates, and you do see a little bit of peak doubling, and there are some examples of that, and I was trying to generate data on that, so I have a bunch of examples. I didn't look into it in more detail. From my experience, the chiral resolution on the column is not as good as some of the other columns. So if anything, you'll get a little bit peak doubling. I don't remember seeing anything where I got some distinct doubling. And, I would just report the average of the two, just one number.
Okay, so in some cases, I actually have seen, especially for macrocycles, pretty significant, when they're racemic, pretty significant change in retention time. And I was wondering do you report two values? Yeah, the question was, is it an average or, yeah, that was kind of the part that I was confused about. So thank you for the thoughts. I think it would be a worthwhile study maybe to see if it does correlate to eventual permeability, if it is related to some sort of intramolecular hydrogen bonding for that particular racemate. It'd be worth looking into.
You'll have to clean it up and to separate it first, I guess, and then test it separately in order to have your answer.
That's a really good question. Any other questions from the panel while we're here? All right. If not, we'll move over to Romulo. You want to go ahead and share your screen and
Absolutely. Well, I want to first thank everybody for attending, and I want to thank you guys for your time. I want to thank Gilles for this great assay, which has been a great contribution to drug discovery. Can you all see my screen now? So I want to start by saying that I call it an unexpected assay because even though it's something I had never heard of, I was first introduced to EPSA by John Riley back when I was at Novartis, and I thought it was really interesting. And then in 2015, I met my former lab partner, Jim Horstick at AstraZeneca, and he's really was the force behind using the shorter method, which I'm lucky enough to have worked with him to talk about. We started developing it, and in 2018, we did a cross-site rollout about all the AstraZeneca sites. And in 2019, it became a routine assay for all wave 1 compounds. The main contribution I feel that we have to this conversation is the shorter method. As you can see here, we've reduced the runtime from 16 minutes to 4 minutes. And on my next slide, I'll have a little bit more details. And by working with the other sites, we decided to start adding QC standards to every run, to every set of runs, so we would have good, reliable data. And we enabled automated processing by using mass links, open links, which will give us a report. So what you do is you run your standards, you run all your compounds, and then it generates an Excel spreadsheet. And then using the calibration, we can just generate the numbers. It's very quick. Some of the useful tips and caveats that I have observed over the time is definitely I would recommend considering using the shorter column and the faster method, especially if you're trying to do many compounds. Or your 8 calibration standards can be run in one injection. The way we would do it at AC, we would run it in quintuplicate, and you will see that these standards, they tend to be pretty right on. You don't see a lot of variation from one run to the next. And if you start seeing variation, there's something going on with your system. You should be watching the retention time of later eluting peaks for column health. I noticed that after several hundred injections, the later two peaks will start to decrease in retention time. That's a clue that there's something going on with your column, and you definitely should consider replacing it. If you want to, keep the column around a little bit longer, you might want to take it off the system, flush it with just methanol. I honestly didn't bother. We were doing so many compounds that after about 3 months, fine, we just throw another column at the system. It was no big deal. Using SFCMS can help you automate the processing, obviously. And if you're doing critical comparisons, if you have a set of compounds that you really care about the relative differences in EPSA, you should run them all at the same time. Even though the retention times tend to be very consistent, you want to be able to compare apples to apples. I would suggest you run them all at the same time. If you're doing method transfer across sites, write standard protocols. One of the mistakes we made at AstraZeneca is, you were using just slides from a presentation and not everybody was using the exact same conditions. Make sure you have a protocol well written when you start transferring it across sites. In summary, we definitely using the shorter method will give you a 4 times faster throughput. At AC, we started with small molecules, but about 63% of the samples that we were running were beyond rule of 5. So there is certainly room for exploring beyond rule of 5 compounds. Sometimes EPSA can predict permeability better than just the 2D calculated descriptors like, TPSA, and EPSA is very sensitive to 3D conformational effects for small and large molecules. What I'm trying to say here, if you have a matched pair and you see a difference in the EPSA value, you should definitely look at the NMR and start considering whether there's conformational changes. And here is, some details on the fast assay conditions. Again, these were developed by James Horstek. Same column, we're using the Kyrex 3014. We went from a 4.6 by 250 to a 4.6 by 50 millimeter column. Here's our streamline method. We're doing a gradient, 5 to 60% gradient. We did here's our correlation of the standards, and we went on and we did a lot more comparison, a lot more validation. We took sets of compounds, running with the long assay, with the short assay. We saw really good correlation. The shorter column enabled us to do a quicker analysis, and the faster gradient enabled faster throughput. I do have one more slide that I want to show. This is a comparison of using the short method and the long method. Again, the numbers are not exactly the same, but you see that the relative order remains the same. Compound 139, retains a little bit less, has a lower EPSA than compound 168. That's really all I have. If there are any direct questions, so we could move my questions for later on, that'd be great.
No, actually, let me see here if there's any questions for.
Okay, I have a question for you.
Yeah.
Do you use UV or are you using mass spec as your detector?
I'm using both. You can do it with just UV. The problem is a lot of times we would get these compounds from the compound library and there would be multiple peaks. The advantage of using, UVMS is that when you process it using open links, it'll report the peak that has the mass of interest. So right. For example, it would run say 80 compounds, 75 of them I would have an exact match of the one that it was there. So the nice thing about that is I even had an idea to run EPSA on crude compound mixtures because we're doing chromatography. So it doesn't have to be pure. But the mass, you're going to know which is the peak that you're looking for. I didn't generate enough correlation data between, mixtures and pure compounds to be able to say, yeah, this is the way to go. But definitely having mass spec, will help you a lot, especially if you're running compounds that are coming from a compound library or a pharmacy.
These are single injections?
These are single injections, yes. For my actual unknowns, I would do simple injections. For the standards, again, I would run 5 injections with all 8 standards on them, and they would vary extremely little. It's a very robust assay. You get good reproducibility or retention times.
Thanks.
No problem. Thank you.
So Romulo, real quick, I loved your comment about, if you need to just go back to the long method if you want to really tease it out. I've even talked to projects where I've said, you know what, there's really no magic to these EPSA standards. Go ahead and give me what you think are poor permeable and good permeable. I'll run those as standards
Yeah.
And I'll just redo the whole assay, and then I'll give you specificity within your compound class. So, there is some flexibility. It's chromatography, right? So it's really, if you create your own calibration curve, you can just reinvent the assay.
Yeah, the nice thing about this assay, and people need to keep this in mind, is that we're not looking for an exact value. We're looking for relative values of, as long as you're doing the same assay over and over and over, you're just looking for things that are, more or less permeable. Like you're looking for large difference. Let's say an EPSA of 80 versus 120. So whether it's 81, 79, you're looking for the bin that your compound falls in. So yeah, you don't have to get too wound up about getting exact numbers.
Perfect.
Romulo, how important is sample prep that you found when running the assay?
I found
using DMSO stock solutions, for example, versus taking, powder. What type of sample prep have you found?
I think the sample prep is not that critical. I mean, I wasn't using compounds greater than, 1600 Daltons. That was probably the most I ever did, but we will get DMSO stocks, 10 millimolar DMSO stock, and we get 5 microliters, diluted up with a pipette and 100 microliters of methanol, shake it up and inject it just like that. I never had a problem with the DMSO. Again, it worked really well. So very minimal sample prep. We would get them on plates. At first, we would get them on vials, and then when we started getting enough, we just started running plates.
All right. Any other questions from the panel? There's a few building up in the Q&A. We'll get to those in a little bit.
Thank you very much.
Next up, thank you. It was excellent. Next up, we have Huy Nguyen. You want to share your slides?
Can you see my slides?
Yes.
Yeah.
Perfect. So this is going to be just a quick slide for me. I'm relatively new to this EPSA assay. We've been running it here at Genentech for about 1.5 years, and really have ramped up this assay in the last year or so. But kind of what Romulo and Gilles was alluding to, a lot of our compounds are now in the beyond the rule of 5 chemical space, and so one of the bottlenecks that we have, or issue that we have, is cell permeability, especially with macrocycles and these cyclic peptides. And we're trying to tease out sort of the chemical properties that will lead to that permeability space. And so that's kind of how we came across Gilles' paper. This kind of in the middle here, where you have EPSA and cell permeability in the y-axis, and that magic number 80 threshold. You kind of want to get below that number. And from my experience running all these EPSA assays, we've actually found that EPSA alone doesn't generally give you predictive permeability, but it gives you a general trend for a given scaffold. And additionally, we actually use other phys-chem assays, like the immobilized artificial membrane assay, where the column itself is the lipid, the phosphatidylcholine lipids on the resin, and that gives us actually some more additional insight on a molecule's permeability. And then also the log D in 1,9-decadiene from Scott Loki's group. Additionally, that also gives us insight into a molecule's permeability. So really, we take into account all of these assays to assess or predict the molecule's permeability itself. And it really leads to the question I think a lot of people are interested in is, when predicting cell permeability, what other assays are out there that we can combine with EPSA and these other 2 assays, or what other computational data that some other people might have that are currently using to get to this answer. So those are my 2 slides. And, I would like to add to Romulo's insight on making this assay more high throughput. We actually attached a a SciX 5500 mass spec to our SFC system. And I know one of the questions in the Q&A was, can you actually mix and pool these unknown samples and run them? And that's what we're doing right now. We're pooling eight samples together. And you're tuning these molecules so you know the parent and daughter ions, so you can mix them together. And we actually just analyze all 8 samples in one injection. So
Right.
it makes it a lot more high throughput.
That's great that you mentioned that, because that was one of the questions that was coming across in the panel, in the Q&A section. So kudos to that.
Cool.
Sorry, I hit mute. All right, very good. Let's move on to Erin. You should want to share.
Sure. I also have just a very simple slide, and I apologize. And I also do have to say that I'm representing myself and not AbbVie in this panel, so that's part of why it is a little bit basic. But I want to give a little bit of an overview of the method that we're using right now, which is essentially the published method that Gilles had just presented, and looking at it, same type of impact for looking at experimentally derived polarity values, particularly for beyond rule of 5 compounds, like was mentioned in the past. And then we are using the 7 drug-like compounds, and now this kind of almost seems outdated, because what Bill was saying about having a project-based sense of set of compounds actually does sound quite clever to do. But one thing similar to is we are using cassetted, we call them cassetted plates at times to increase the throughput. We're doing 5 compounds per well. One thing I've noticed is that within a DMPK-like organization, these type of plates exist and are routinely made all the time. So, if you're running this assay and like we are embedded in a medicinal chemistry organization, teaming up with DMPK that already has a lot of these throughput advantages has been really useful for me. So like I said, a lot of these will have internal standards. You could perhaps have your own internal standards put in there, like Romelu had said. And since each one is a tuning plate, exactly what's in. You can use, build a sample list that has extracted ion information and then be able to identify 5 at once. We're currently using an Agilent system, the 1260 hybrid SFC system with a single quad mass spec. We like it because, so looking at some of the shorter methods, there is a throughput disadvantage where using a really high flow rate on a long column like this can have, problems with it overpressuring. So that's maybe the disadvantage. The advantage we like is it's quite easy to build automated sample lists from Excel and run calibration curves and have all the data pretty much inputted directly. And if you get pretty clever with having an export into an Excel file, you can have some of this data pulled directly into internal databases, which is what we've been working on. So actually one question I have is, it's if you're storing, might be worth this, part of this discussion, but storing a large amount of EPSA values similar to, PAMPA or E-log-D, where there's just a database of this information and you have values. I'm wondering if that's, the best way to represent this kind of data. So there is the possibility to have that, stored somewhere. But yeah, that's kind of what we're working on right now. So yeah, the focus in our lab is to analyze beyond rule of 5 type compounds, and we've increased our throughput mostly by cassetting plates, being careful not to have compounds. A lot of times within the project, if you're looking at those very specific type experiments, like Gilles was saying, where you're looking at individual, like a gamma versus a beta turn, and they'll have the same molecular weight. It's not an experiment to cassette your plates, but when you're looking at just trends in overall permeability, that can be a really useful tool to increase your throughput.
All right, great. All right, so we'll again we're building up some questions. We're hitting quite a few as we go along, so that's great. Next up, we have Johan. You want to share your screen and?
Yes, let me get that. Do you see my screen?
I still see you.
Oh. Let me go there. That's weird. Okay. Let me try this. Do you see my slides now?
Yes.
Yes. Yes.
Okay.
This came through. Perfect.
All right. Great. Thanks. Yeah, thank you, first of all, for inviting me to this webinar. It's really interesting discussions. So what I want to touch upon today is what we've seen is that EPSA in itself, at least for our compounds, it couldn't predict permeability, and it really depended on the scaffold that we were looking at. So I'm going to share a few data where we related EPSA to permeability for several in-house projects. These are the molecules on which we tested EPSA were not macrocycles. So in that sense, they were different from the compounds that you guys presented on today. So maybe there are some learnings, maybe there's some more information that we need to obtain, like Huey already referred to, is maybe you need to combine EPSA with other parameters to get a better prediction of permeability. So these are just some new data because we are quite new to the EPSA assay. I think we started running this assay last year, so about 2 years, 1.5 years ago. So I think the same time as you started. So first of all, I'm going to show you these two graphs here. On the top graph, so what we did run was we looked at passive permeability, so Kicker2 assay plus a set of inhibitors to inhibit active efflux transporters. And we looked at this passive permeability across different projects where beyond rule of 5 type compounds were synthesized. And what you can see is that at least for this first assay, that this EPSA cutoff of around 90, 95 could indeed be applied, at least for the yellow project. As you can see here, we really got a tremendous increase in permeability as we decreased EPSA. Now, what you do have to realize is that these molecules were very close analogs, as Gil already presented before, so that for close analogs, these small differences in EPSA could really make a difference. However, we were interested in intrinsic permeability, and since this first assay was run at a pH gradient, we therefore switched to a Keiko2 assay where we kept the pH the same at both sides of the monolayer. And again, we looked at passive permeability, and what you can see now in figure B, sorry, let me put that in a laser pointer. What you can see here is that we didn't obtain this nice correlation anymore. Mind that these are not per se the same compounds, but you can see that at least for the yellow project, we could not maintain that cutoff of around 90 or 95, which was interesting in itself. And also what you can see is that there are compounds here with an EPSA of well above 100 in this orange project where the passive permeability even reached over 100 nanometers per second, something of 10 times 10 to the minus 6 centimeters per second in another unit. So this told us that the correlation of EPSA with permeability may even depend on experimental conditions applied in the permeability assay, and that correlation of EPSA with permeability for close analogs could work or may work better. But then for these type of molecules that we were looking at here, maybe you need to combine the EPSA with other physicochemical descriptors. So this is something that we only recently started to try. So that's something that we're trying in this figure. This is something new. I don't know if it will make if it will hold if we get more data, but these are some new learnings for us. What you can see here is that across different projects and also using passive permeability data across different Keiko2 protocols, we rank these molecules in terms of EPSA and that delta log D. Most of you are familiar with delta log D, where it gives you the difference between log D octanol minus log D toluene, and that could give you some information on how likely your compound is to swap between the aqueous environment and the intramembranar environment. And what you can see here is that for these compounds where you get an EPSA of around 130, so well above the cutoff, you still get a decent permeability ranking. So in high permeability range where you're in for these green compounds, green circles here, and that these compounds which tend to have a low permeability, at least for the projects that we were looking at, they tended to cluster together here in this region. So these are just preliminary data, and we may need some more observations to see if this holds. But I think that based on these data, that it's likely that you need to combine EPSA with other parameters, such as the radius, as Gilles already showed in his first slide, and maybe some other in silico descriptors, such as the number of rotable bonds or the delta log D in this case. So that's it. And I also want to acknowledge Anthony Severino, who's actually on the call and who really generated all of these EPSA data, Magdalena Kirkovic, Jean-Marie Nicolas, and Mohamedullah, who've been working on this together with me. Thanks.
That's great. Now, is that, you said that was Keiko-2 permeability data?
Mm-hmm.
Okay, so yeah, so one of the things that we had spent some time on was to try to avoid any kind of permeability that wasn't just passive, right? Like we really got to avoid, you know, I think we tried to eliminate Pampa and Keiko because they also add some other mechanisms of permeability, reflux and such. So that just makes my life, it's too confusing for me to work through. So we tried to pick the RRCK assay because that at least is mostly driven by some sort of passive permeability.
Yeah, well, these data were obtained in Keiko-2 assay where we co-incubated with efflux transporter inhibitors. But also when I looked at these compounds or other types of molecules on a similar type assay to the RRCK, which is the LLCPK1 assay, I will get similar results.
Well, it's good. I love to see the progression of the science here.
I have a question for Johan.
Yeah, go ahead.
What was the rationale of making a pH gradient in your passive permeability assay? I wasn't quite familiar with that trick.
Originally, we were looking at intestinal permeability, and so applying an apical pH of 6.5 would mimic the average pH change across the human intestinal system, and that could give us and some information there. And we were actually modifying our assays to better accommodate the beyond rule of 5 type space where you can have stickiness, etc. And also because we wanted to look at intrinsic permeability, because there were some questions around whether some of these molecules would actually enter into the cell, into our pharmacology assays, where obviously you wouldn't have this pH gradient. So then we returned to the intrinsic permeability assay, where we kept the pH the same on both sides of the monolayer.
Okay, yeah, because when I've thought about the gradient that occurs between the stomach and the end of the intestine, I mean, you have basically every single pH almost represented, and as soon as you are in the right conditions, you will get something through potentially. And it's always, in my opinion, it was always very tricky to take the intestinal absorption at face value, because we have a model that does it in a certain condition. Because in a real body, I mean, you will have all the different pHs present. And if something is permeable at one of those pHs, it's going to go through no matter what. And that makes it even, I mean, that makes it really tricky to predict and to model in a lab, essentially. Just some thoughts.
I have a question for Johan too. And, looking at your relative permeability data, I'm wondering if you looked at it at all. You have permeability, like you said, in the intestine, but how does that, in the context of oral absorption tend to look? Like if you're looking at oral bioavailability for some of these compounds, does it, if you bin it, within those types of, good, good absorption versus bad, if your data kind of can be, I don't know, massaged a little bit different, not massaged, but look a little different in that case?
So these are very early projects, and I currently don't have oral bioavailability data yet. But what we do have is that the first assay was really, it was calibrated for oral, at least fraction absorbed. And we do have really good cutoffs in terms of oral absorption there, which tend to hold now. Indeed, like you say, it may be that these cutoffs that they shift for these type of molecules, but unfortunately I don't have those data. That would be really interesting. Yes, I know that there it could be that, and I think that there are some papers where lower cutoffs are applied for oral bioavailability for these type of molecules. So that may mean that we could look at lower permeability values. Yeah, thanks for the question.
Johan, was mass balance an issue for your permeability assay?
No, we specifically looked at this, and then we tweaked the assay in a way to counter some of these observations. But what we did see is that even if we ensured that mass balance was okay, like let's say above 75% recovery, that even after tweaking it, so even if it was like 50 or let's say even 40% recovery before tweaking the assay, that after modifying the assay, the passive permeability values wouldn't really change. It was only when you started to go into recovery issues at a value of below 20%, let's say 15% recovery, then your permeability values tended to change when you improved recovery afterwards. Thanks for the question.
Well, any more questions for the panel here? Before I move on, I want to do one last presentation, and then we will move to the Q&A. Just give me a second here. Okay. Going to share this. Can you all see that? Yeah. Okay. So today, the panelist Paul Lefebvre will be played by me, and we'll go through his presentation. As those of you that joined the call late, Paul is expecting a grandchild today, so he's off tending to more important things. So I did want to cover our host today and Verica Discovery Services and what they offer. Way. Okay. Who they are and what they do. They're a boutique analytical development CRO with extensive experience in small molecule chromatography, analytical and preparative. They were established in 2007, located in Marlborough, Massachusetts. They're globally serving clients in pharma, biotech, agriculture, chemicals, veterinary medicines. They have a reputation in SFC. Their reputation in SFC led to development in other analytical areas like impurity isolation, identification, method development, phys-chem testing, and etc. What they do is they help scientists who are struggling to find adequate high-quality material supply for their development programs in short time, chiral, achiral, and bulk purification. In a rush to separate or identify compounds of their samples, including impurities, API, and other degradants. And those that are in need of reliable predictive phys-chem data like EPSA, log D solubility, so on. They do perform the EPSA assay in-house, and they use the same instrumentation and expertise that from Gilles' published method. And the contact is our host, Paul Lefebvre, and I've left his information there. We will, maybe I'll leave this up for a little bit as, oh no, I can't. Oh, I think I can. Okay. Okay, so I'll go through some of the questions that have come up and then open it up to the panel. I will try to move between the Q&A and the chat function, as some questions have come there. So I'll start here in the Q&A, and then we'll go from there. First question, in what type of compounds are EPSA most utilized? Small molecule, large molecule, peptides, small and large, and why? Anyone want to take a stab at that?
I'll try. We mainly use it for larger molecules. And I think the overlying problem that we have is trying to predict permeability. And I think the EPSA assay does a much better job with these larger molecules.
I'm gonna second that. I mean, we're working on a lot of PROTACs, which were medium molecular weight, around 1200. As I said, about most of the compounds had at least one rule of 5 violation. So I would say medium molecular weight compound projects were the ones that were more interested in generating the results for us.
That actually leads me to another question that had come in, is what's the maximum molecular weight you've been able to study with EPSA?
Personally, I've gone up to 1600. I mean, but that's just what was coming in for us.
Yeah, I think you're about right. I think I've seen things coming through going up to the 2000 and more, but most of the time those were way too polar to have an illusion within the regular gradient, and obviously we're generating issues with the mass spec as well, so double charged and things like that. But I mean, 1700 is a very reasonable top cut for correct illusion within the gradient. Because you also need to remember that once you are in the rinsing period of your gradient, you're basically flushing out everything that is stuck to the column, right? So
yeah.
If maybe I can also still contribute to the previous question around where do you use EPSA for which type of molecules? So it depends obviously on what the endpoint is, because we did try also for compounds which were within rule of 5. And if you then related that to permeability, we didn't see, at least for the compounds that we were looking at, we didn't see any correlation there. But what struck us most is that those molecules were not flexible at all. So there may be something that you need to consider in terms of flexibility, but I think that Gilles already showed at the beginning of his presentation that for the smaller molecules where you did have this intermolecular hydrogen bond, that it may work. So it may help to discriminate, but then to have these larger data sets where you want to have a single cutoff there, I think for these non-flexible molecules, you may struggle.
yeah, I agree with you, Nico, Johan, that we've seen more impact on larger molecules, on cyclic peptides and inflexible molecules like you just described.
All right, so another question here, and maybe this is focused for those that have tried to ramp up the assay. It says SFC measurements in the standard method on the 250 millimeter column compared to the short method on the 50 millimeter column, showing about a 10% lower EPSA on the short method than the long method. And they're wondering if we had seen a similar trend.
Not really. When we did our validation of the shorter method, they were pretty spot on. That's all I can say. So no, I had not seen that. I mean, the numbers won't be exactly the same, but 10%, that seems quite a bit. So I would suggest you know, check the grade, check your gradient, check your back pressure, do your basic checks to see what's going on. I have not observed that.
So,
To add
Romolo, if
to-
you were going to switch from using a longer method to a shorter method, you'd be able to report all of those values collectively, or would you recommend rescreening everything?
It really depends on how much you're talking about. I think that what we saw they were pretty spot on. I don't think there's any need to rescreen everything, except maybe, let's say you have, again, a set of 10 compounds, a few you had run earlier with your longer method, and now there's interest in them, and then you want to compare them apples to apples. Yeah, don't rescreen everything, just rescreen, things that you might have interest in.
To add to Romulo's answer, we also tried a shorter method when we first started this, because we were thinking 16 minutes, that's way too long. And I observed a very similar thing to what the question was, where there was a 10% difference or more between EPSA value. But I think the key is you have to change the flow rate and make sure the pressure is different for the shorter method. Otherwise, it won't adjust.
Interesting.
We're keeping the same flow rate, and it just, yeah.
Okay, interesting. Thank you.
Yeah. Okay, that is interesting. The simple question is: the ammonium formate mandatory? Anyone?
Let's
Gilles?
put it that way. You don't get the same results if you don't put it.
Yeah. I think it was just to have enough buffering, like counter ions in there so that you don't get extreme retention, extreme interaction from one group or another. I noticed a little bit in the beginning that there was a little over retention of carboxylic acids, very strong acids, sulfonic acids, and so then adding something to kind of counter that made it a little bit better.
And also, once the method is established, and if you want to compare apples to apples, you stick with what you have, and it worked for us. So we didn't need to change anything.
So I know, Aaron, you mentioned that you were using the Agilent. Gilles, I believe you're Agilent. I'm Agilent. The question is, what SFC are you using? So I open it up to Huy, Romolo, and Johan.
We used UPC Square. We had 2 UPC Square systems. Actually, across sites, we used UPC Squares. So it was very, very transferable.
Okay.
We
Johan,
use,
go ahead, we
use Shimatsu SFC system.
Johan?
For us, I just asked my colleague. It's the UPC squared system.
Okay. Okay. This question here, how many compounds? I think it's for, actually, I don't know who it's for. How many compounds have you analyzed by EPSA, and what have you compared it to test its accuracy?
I think last count it was about 5,000 we had done. And then for some cases, we did compare it to Keiko2 data. And we found out that not for all compound classes, not for all projects, it mattered. But if there was this one particular series, pyrimidine compounds for this one particular compound, we found that the EPSA data correlated really well. And what that allowed us to do is we were able to decide which compounds to send for Keiko2, because we don't want to send everything for Keiko2, but compounds that had promising EPSA data, then we were able to, okay, let's send these for Keiko2. So we have that value.
I think you overtook us in terms of volume. I think we're around 3,500 last time I checked.
So let me see. The latest is about 7,000 compounds as of July of 2020.
They're blowing us out of the water.
So that's the difference between a 4-minute and a 16-minute method.
There you go.
All right. Okay. So another interesting question. So when analyzing several compounds in one vial, what about influencing compounds? What about the influence compounds might have on each other and building intermolecular hydrogen bonds, or even, I guess, just pairing up, I guess is maybe the answer. Any thoughts on that? Anyone?
We actually did this study when we were trying to cassette our samples, and we tried pooling acids and bases, different charges, and we actually didn't see any changes in the EPSA values compared to just single injection.
I haven't seen it different compared to single injection either, but I haven't done my due diligence to test it either. So everything. So yeah, if a concern, I would run them separately as well. And for the Agilent system, they have to be the mass, the exact mass of each one has to be, within m/z of plus or minus 10. Otherwise, you're also quite in a confusing range too.
Well, the only one that are mixed in our case are the standards. So we pull all the standards together and only do one injection instead of 7 or 8 or 9.
Yeah, likewise.
Yeah, we wind up running out of plates, so each well is its own purified compound or clean compound anyway. I don't know, we'll see if this is in scope for here. Is how critical is it to run these EPSA experiments on SFC versus, say, UPLC? Any ideas on the role of CO2 versus hexane or heptane? Anyone?
Well, I think it's certainly in terms of epsilon values, you won't get the same pristine dielectric constant using an heptane mixture. And I mean, you have all the environmental component as well that you need to take into account. And I mean, normal phase is not the most liked method typically around the industry, even for purification purposes, but also in analytical area.
And I don't think you'd be able to achieve the same flow rates either. So I think you'd be able to negatively impact your throughput by switching. So for me, that was the biggest. It's already a long method, like I said. So yeah.
I had never actually tried it myself in the heptane. I just had made the observation that it appears that those that have intermolecular hydrogen bonding elute first in SFC, and it was irrespective of what column I was on. And so it just stuck with it. Again, would not go down a hexane path for environmental health and safety reasons. It's not very green either, right? Aaron and I serve on an ACS green chemistry team, and we would be shying away from hexane for sure, and heptane as even an alternative. So okay, done. Okay.
Well, I guess,
has anyone tried to use the Chirex 3014 for preparative SFC? I don't think that's really in scope, but I'll throw it out there and see if anyone has an answer.
So
I haven't. I can't. I'm not even sure you can get it in preparative.
Yeah, I was wondering, do they come in fat columns? I'm not even sure.
So did you ever face issues with establishing the correlation for any chemical subseries?
I mean, that would depend on what you want to correlate against. Are we talking permeability or? And again, you not necessarily want to have a one-to-one permeability EPSA. What we found is that low EPSA is necessary, but it's not the end all be all. I mean, without low EPSA, forget it. But with low EPSA, below 80, let's say, or 90, depending on the series, you increase your chances of being permeable dramatically. And that's where we basically took it from. It's just hedging your bets and making sure that you are in the right area and saving your resources with not.
So maybe this will help, because I see another entry. Can the panel comment a bit on carboxylic acids? These groups often contribute an outsized amount to EPSA values. I think means they're a little overexpressed in polarity. How do we think about pKa in the supercritical fluid environment? Are EPSA values sensitive to pKa modulation? If so, why? So maybe that's the let's see if maybe that's the question. In terms of correlation, maybe it has to do with the acids. I'm checking the chat panel to see if maybe there was a comment put in there to this. I don't see it.
Well, acids have a tendency of should I say streaking or hogging the stationary phase quite dramatically, and it's easier to compare a series of acids together rather than trying to compare across neutral bases and acids. At some point, we try to develop a specific acid type method, and then all of a sudden, nobody was making any acids in the company anymore. So it wasn't a priority. So we kind of let that on the side. But I mean, there was an effort at some point to develop something acid specific, where we would compare just series of acids and not. I mean, it would have been an EPSA for acid assay, not a classical EPSA.
Next question. Oh, go ahead. Go ahead.
I was wondering if people at AZ or ABI were exposed to acids and how they were dealing with them.
No, many that I can recall specifically.
Yeah, none that stand out. I mean, I think they have been in the mix, but I haven't actually gone through to actually pick those out and see if as a trend, overall that those exhibit different properties. I did also note in the literature too that has been commented on some of in the chat that they were shied away from as well. So yeah, it's definitely something to take a look at, because they typically are late eluders as a category. And in that case, thinking of it in terms of how Johan said, like using it as an adjustment value in collaboration with other DMPK assays might actually highlight some of that differences.
Anyone else?
Yeah, how would you even, I mean, you measure the EPSA of acids, but how do you know that's the wrong value, right? Or if that value is inflated? Like, the only thing you can really compare it to is the TPSA. It's hard to compare whether it's the right or wrong answer.
Or you take it to a permeability assay and you then create a whole new correlation, right? Oh, and this is an acidic compound, and and you could actually redevelop the method specifically for acids. I think, Gilles had tried that, but then no one requested it. So then what?
Yeah.
But you could, you know, I don't think there's anything stopping you.
Well, this is an interesting question I have for everybody. How do we get this assay more popular? How do we get more buy-in from medicinal chemists? Because this is one of the things that Jim ran into at AZ. You know, it took years for them to even start getting some interest. So how do we get people to recognize the value?
By running events like those, probably. That's a good start.
Yeah.
Well, I think, you know, just to use Gilles' data as an example, is when you have nothing, and then all of a sudden you have something, right? So he has all these peptides that just don't correlate anywhere. These guys are shooting around in the dark, trying to figure out how to make this more permeable, but yet you come and here's an assay that actually helps the design of molecules, leads you in the right direction. All of a sudden it becomes very popular. I think you just need those kind of case examples to really drive the assay further and further into the design process.
I think it really takes collaboration between the DMPK department, the medicinal chemistry, and the SFC department just to try it out. We're all scientists, so if we see something new, then we want to try it out. So and if it can help, I think I agree. Just generating those case examples.
Here's one, Gilles, from one of our good friends. What's your experience with applying EPSA to improve the permeability of PROTAC compounds?
We basically have held off of the PROTACs right now because PROTACs can be injected as well, and they're typically the lowest hanging fruit in terms of first you want a PROTAC that works, that does what it's supposed to be doing, and if you really want to add another complexity level of making it orally bioavailable, you're in for a longer development time, a longer research time and resource investment. So I think the PROTACs, the first generation of PROTACs, will probably going to be parenteral formulations rather than pills, and because one of the issues is that you have so many moving parts in those compounds. I mean, and if you're making an intramolecular hydrogen bond between the E3 ligase portion and the warhead, what tells you that it actually is going to stay there once it goes through the membrane, and actually not stays there and shoots you in the food, depending on the microenvironment that compound gets into. So, and PROTACs is a completely different ball of wax, to be honest.
Okay. Any other comments?
On this question specifically, maybe that we did also try a few of these PROTAC-like compounds to see if our compounds did behave similarly, and I think they're if I then connect with what Gilles' observation was, then indeed that could be the difference between the microcycles and these longer flexible molecules. There may be some differences in how they behave, like you said, but that would be really interesting to see what really the descriptors are that can be added to EPSA to tease that out.
So actually, you know, cycling back to the acids question, and there's several questions around here. So, you know, maybe the one that maybe I'm going to focus in on is anyone researching this further? EPSA for more acidic molecules. That seems to be the basis of what most of the questions are. Anyone? New opportunity for someone out there?
I would say most of the molecules are on the basic side or zwitter ions.
Yeah, like I said, you have to have the need to invest the time in developing something specific for acids. And when the pressure was there, we worked on it. And as soon as the pressure released, we had other fish to fry and more important deliverables. So, I mean, yeah, I mean, everything is cyclic. I mean, there's going to be new targets that will require carboxylic acid moieties to have reasonable potency, and EPSA might have to change its way to make it work better for carboxylic acid if the need arises, and that's always the need in the question. Do you really need it, or is it just a nice to have?
Oh, here's a good one. What would you say is the biggest limitation of the EPSA assay right now?
Instrumentation. Not everybody has SFC, I would say. Although you have places like Averica that will provide it for you, so that doesn't necessarily stop you.
Correct, right? I'm sure they're happy to hear you say that.
No, it's true. I mean, that's the problem with SFC generally overall, right? Validating methods by SFC, it's a complex technology. So not everybody has it. And the fact that Averica would provide it, yeah, dip your toe in the water and see how it goes. Don't let it stop you, the fact that you don't have an SFC.
Too, and this relates to the buy-in for medicinal chemistry, is there's just a value is contextual, and you can't just report a value and have it have a specific meaning. And there's kind of cutoffs for permeability, but really they have to be individual. For one therapeutic area, this is, a study that we ran, and we rank order these compounds is kind of a better way to look at it. And so I think a lot of times when we start to see a fraying of the data is when we pull data from, thousands of compounds across different projects, and then your bins look a little bit odd. So I think that is maybe the biggest part about buy-in and things still to understand about it.
It's interesting you say that, because one thing I got was as a comment from someone Gilles and I know very well, have a computational model. As if people have a in silico tool that they can just try in the computer, they'll try it and then they'll see, and they'll build some sort of correlation and go, oh, well, I'm doing log P or log D. Oh, look, here's this EPSA. Oh, this looks like it's correlating better. And then next thing you know, they're trying to confirm those in silico results.
Actually, I have a question to Gilles about the computational work. For your model, how big was your training data that year? I guess you had like 3,000 data points or something.
No, we had much less than that. We started with maybe like 1,200, something like that, if I remember correctly. I mean, I was only supplying the raw data and the structures. The whole big black box of generating the descriptors and doing the QSRR in the backend it's like I said, it's more like a black box to me. And I had an expert that did it for us, and that's how it came about. And all I know is that the outcome was terrific, and that we had like a one-to-one with a few exceptions, and sometimes the acids were those exceptions. But as soon as you have like, I don't know, maybe 500-600 data points, you can start having a really reasonable computational model. And we're fortunate to be in large enterprises or large research organization where you have access to expertise like that and computational chemists that are up to the challenge, and they can whip you a computational model quite easily. But the drawback is that you need the raw data point first in order to have anything to work on. And unfortunately, the industry is not very open to sharing structures and data across platforms and across competitors. But I mean, that's the drawback of one of the EPSA, is that everything is proprietary and everybody will have their own model, and comparing across models might be a little challenging, but I don't see why not, because essentially it's just taking the structures and then attaching it the value, and the values I'm pretty sure will be very consistent across all places that are generating it. So I think the proof is going to be in the pudding.
I think that's a good point. Like if we were to provide the model to the public community, I think that would increase the buy-in for this assay.
Mm-hmm.
Mm-hmm.
Right, because you're not necessarily disclosing structure. Like you say, it becomes a tool essentially. Yeah, that's a very good point. Yeah.
May I ask a follow-up question to that? When you say those 1,200 to 1,500 compounds, were these mostly macrocycles again, or did you also include more longer and more flexible compounds in that data set? And does it then, as you just said, so for instance, for PROTACs, EPSA, you get different values. So how does the model predict for those more flexible and longer compounds?
Well, as a matter of fact, we started mostly with small molecules when we built the model, and lo and behold, when we predicted larger and cyclic peptide, they were just finding their way on the line. It was quite interesting, and I mean, not necessarily unexpected, but it was a good surprise to see that the larger molecule were actually behaving and falling on the line. So that gave us quite confidence into the quality of the calculation and the modeling that was being performed.
That's interesting, right? Because you would expect that it's a 3D conformational behavior, whereas you would input a 2D descriptor. Or did you input also 3D descriptors into the?
Yeah, 3D descriptors were a part of the modeling. Yeah,
Okay.
there were extensive minimization of structure behavior in 3D, and I think it was, if I remember correctly, it was modelized as if it was in chloroform environment. So something that was favoring the intramolecular hydrogen bond the way SFC does in the column environment.
That's interesting. Thanks.
Okay, so give me a second here to switch to the chat function and see if I can pick up some. So there were a couple of questions around the assays themselves. Pampa, Keiko, I saw another one here. LLC, PK1, MDCK2 cells, and trying to compare, or, in one case, even asking, is there a better correlation between different types of cell lines and passive permeability? Is there any comments on that to the panel about, which assay to use, which assays you've correlated to, or anything in general that is guiding your research?
I can maybe start the answer of that, answering that question. Although we haven't tried too many compounds across different cell lines, we do have several data. What we recently observed was that, at least for our compounds, when you enter into that low permeability space, then it is known that the membrane composition across the different cell lines can differ. And then the interaction with the membrane could, I think, at least based on the observations that we had, could change. And that could change at least the ranking of some of these lower permeable molecules. Although overall, we do see for the, let's say, moderate to high permeability compounds, we do tend to see a good correlation. So I think there may be some cell line dependent behavior, but that was an observation I made for our compounds. I cannot speak for other types of compounds.
I did have another question about water. I sort of lost it a little bit. How much water, what's the max percent water in the peptide samples?
I wouldn't know, to be honest, because we would get our peptides directly from our sample bank, and they are typically stored in 100% DMSO. So, and I mean, typically the DMSO used is also anhydrous, so on the low end.
Do you know the difference between, there's a large difference. Okay, so this question here, do you look at the difference between EPSA and TPSA? The first question. A large difference, how large, twofold, could be an indication of flexibility. That's right.
Yes, we do. I mean, that's one of the questions, that there are three indications that there is some conformational interactions going on with a molecule, especially if you have two matched pairs. Like Gilles showed, the very first example, they have the same TPSA, but you see a significant different than the EPSA value. That tells you that there is some intermolecular hydrogen bonding going on in the molecule.
If I could also add to that answer, what we observed as well was that when you have these high TPSAs in the range of 200, 250, then I think that the subtlety in differences between EPSA is much smaller. So if you have EPSA values of 100 or 80, and that value is around 20-fold difference, but then the difference in TPSA, which is much larger, that you won't be able to correlate the TPSA minus EPSA, at least in my case, with permeability, because your TPSA is, let's say, 250 and a minus 100, and you get that 150 there. But then if you would then have the other one is 75, then the difference is only like 5 between those two. And so it becomes difficult then to correlate the TPSA minus EPSA. At least that's what I've seen, but maybe you had better experience with that, Romulo?
I'm sorry, my attention was elsewhere. Can you say it again, please?
Did you have so I was saying that if you have these matched pairs, for instance, and you have compounds with a TPSA of, let's say, 250, and then your EPSA is let's say 79 or 85. So we're in a good range. So let's say your EPSA is 85 to 95, right? Then TPSA minus EPSA is very close to each, or could be very close to each other for these close analogs. So then trying to rank TPSA minus EPSA becomes difficult, because you end up with values that are all within 125, 126, 127
I see what
area.
I see what you're saying. We didn't do that thorough comparisons. It was just an observation that if you have these two molecules, that have the same TPSA but different EPSA value, you should look at the NMR and such. But we didn't do an in-depth analysis of it.
Yeah, and to add to that, the more you generate EPSA, the less you rely on TPSA.
Yeah, I agree.
And you basically move away from TPSA altogether, especially if you're in the large molecule world, because of the flexibility in essence.
Yeah, we did find that for us, you know, EPSA predicted better permeability than any of the other descriptors, any of the other calculated, you know, descriptors like TPSA, etc.
That's a good point. I mean, a lot of these predictive models use TPSA as a descriptor. You know, it might be good to just remove that and move to the EPSA for your descriptors.
Particularly once we start developing tools to do calculated EPSAs.
Yeah.
I mean, molecules are not flat, you know.
Yep.
I had one question here, which is, do you think an SFC method to determine lipophilicity, like on an C18 column, would also provide interesting information on permeability? No one?
Sorry, what was that question again?
It was really, can you use the C18 column to determine lipophilicity, similar to you do in HPLC? Is that
that's log?
Is that
that's I mean that's
yes,
that's
log D.
Yeah, that's log D.
So yes, the answer is yes.
Well, EPSA. Okay, so the question here was, will the EPSA of less than 80 for good permeability will hold only for compounds with C log P less than 1, or will it also be helpful for compounds with C log P greater than 4? EPSA less than 80. So I guess it's the where you've drawn the line, Gilles. Is that good for compounds both below 1, C log P below 1, and C log P greater than 4?
Yeah, that's a good example is, for example, cyclosporin. You know, cyclosporin has a log D of 4.9 and a TPSA of 279, but when you look at it, it's, you know, EPSA value, it's
like a 110.
Yeah, it's very absorbable, so which is a good thing.
It's we already know bioavailable, right? Like it moves through the membrane. Yeah, it was one of the early examples of you know, what they were after, is beyond rule of 5. Yeah.
Right.
Well, so
yeah.
I have a very long question. It looks like it's okay. So bear with me. Methodologies. Okay, so the question is, they were under the impression the EPSA methodology is fast SFC technique that attempted to measure embedded polar surface area of a molecule helps or hinders their permeability through membrane. We're seeing here today that EPSA is a useful technique, particularly for large molecules, say those above 500 molecular weight, and measures conformational flexibility, which does not really address the phospholipid interactions. Is that a misconception or a good takeaway from this presentation?
I feel like those are two different things.
Yeah, I'm not understanding the question.
Okay.
I mean, I think, yes, you don't have, in EPSA, you don't have the phospholipid interactions, but that's where the other assays come in, like the IAM column and things like that. But I mean, I think those are, interaction with the phospholipids is a different question compared to the exposed polar surface area, I think.
Agreed. So this sort of like using that IAM column for, isn't there an HPLC assay using IAM that predicts, I think, log D or something. Pretty sure there was an assay like that.
Yeah. Yeah.
Maybe we're coming to the end, folks. We've got about 10 minutes. Still have quite a few participants. All right, so I got a couple of questions, that maybe we haven't covered. Has EPSA been successfully related to other DMPK parameters apart from permeability? For instance, drug metabolism or transporter mediated efflux?
I haven't looked
Anyone?
into that personally.
Oh, okay.
I have a few examples in which and this is part of a talk I gave at SSC 2019, which there is some correlation between EPSA and MDCK efflux ratios. I just have a few examples, not very extensive, but yeah, we have done some of the comparisons. And it does seem to correlate, lower EPSA values correlate with lower MDCK ER values.
I think we have a limited set as well when we looked at the matched molecular pairs, and the ones that were generating intermolecular hydrogen behaved significantly differently than the ones that were not, in essence. Yeah.
Here's one interesting question that kind of came up. Do you observe more than one peak for macrocycle peptides? If so, is it related to multiple conformations for these compounds? Anyone? No?
We do it, for my case, for macrocycles, we do observe sometimes multiple peaks. Whether it's because it's a mixture, I'm not sure if it's different conformations. It's probably a racemic mixture.
Yeah, most of the time we see two peaks is when we actually found out that the synthesis of some of those peptides were performed using racemic amino acid instead of specific ones. And I mean, that also helped the team realize that the QC of the supplier was kind of faulty. It was like an added benefit of using EPSA on a chiral column using a Perker base.
So I did receive a clarification that Clara Valco at GSK developed an IAM assay trying to tease out when compounds bind to a membrane. So just wanted to give a shout out to whoever sent that. Thank you for that.
It was John Riley.
Okay, any preferred scaffolds for generating cell permeable macrocyclic peptides, cyclosporin type and anything else? What about stabilized alpha helicopeptides? I'm not sure I can answer that. Anyone? I'm not sure that's in scope. Yeah. All right. Sorry. We're gonna have to.
I would like to know that answer too.
Yeah. Right. So great question. New research project. All right, one last one. How much percentage of EPSA data can match with Keiko from your experience in terms of permeability? So I think we sort of answered all that. Okay. Well, folks, I think we have come. You guys want to, any parting comments before we close our session down?
Thank you for hosting this. Yeah.
For hosting, yep, for inviting me. Yeah, got a lot of burning questions I had answered, so thank you.
Well, really, it's thanks to you. for coming and sharing your experiences. And thank you, Gilles, for putting this together and leading the way and getting this assay out there, and all the hard work you and Simone had done with building the model and putting it into place. Awesome work. Awesome work.
Yeah, maybe one day we'll have a global multinational model available for everyone. Who knows?
Just like an automated, commercially available, Abrams predictor package of some sort, right?
There you go. Yeah. Mm-hmm.
Yeah, perfect.
And with Averica available to generate the data, and there's the sky is the limit in essence.
Well, I think that's it for us. Jake, anything you want to finish up with? You want to close us down? I don't even know if he's still there.
All right, guys. Thank you all. Good
All
to
right,
see
well,
you all. You look great.
Why don't I release you, and then we'll close all this off. All right. Well,
All
thank
right.
you all.
Thank you again.
Thanks for nice
Again,
discussion.
this will be available on the Averica website, and I wish you all the best. Stay healthy and stay safe, and have a good morning, afternoon, evening, depending on where you are in the world.
Thank you, everyone.
Thank you,
Bye-bye,
All
guys.
right, thank you.
Cheers.
All right.
Bye-bye.